My name is Junhyung Cho. I'm an assistant professor of molecular cell and systems biology at UC Riverside. My lab studies the neural mechanisms of fear learning and memory.
My name is Eunbin Kim. I'm a postdoctoral researcher in the Cho lab.
In this video abstract, we'll be discussing the synaptic mechanisms of discriminative fear learning and memory. To survive in a dynamic environment, animals develop fear responses to dangerous situations. For these adaptive fear, animals must discriminate between different sensory cues and associate only relevant stimuli with aversive events. To investigate the mechanism of discriminative fear learning, we trained mice to learn to associate an auditory conditioned stimulus or the CS+ with an aversive unconditioned stimulus, such as a shock. After discriminative fear learning, mice show fear responses to the CS+, but not to irrelevant stimulus or the CS-. Both the auditory CS+ and CS- signals are conveyed to the amygdala, a center for fear learning and memory. Long-term potentiation, or LTP, in these pathways plays an essential role in auditory fear conditioning. However, it was unknown how LTP contributes to discriminative fear learning. We hypothesized that LTP is induced selectively in the CS+ pathways when mice learn to discriminate between the CS+ and CS- and show fear to the CS+. To test this hypothesis, we examined how synaptic strength in the CS+ and CS- pathways changes after discriminative fear learning. For this, we labeled the CS+ pathways with channel rhodopsin, which is activated by blue light. Blue light illumination in the amygdala selectively activated the CS+ pathways and induced the synaptic responses. With this approach, we first examined how discriminative fear learning affected synaptic strength in the CS+ pathways. Here, mice in the fear conditioning group learned to fear to the CS+ but not to the CS-. The AMPA-NMDA ratio, a proxy for postsynaptically expressed LTP, was significantly higher in the fear conditioning group than in the no-shock control group. In the CS- pathways, however, there was no significant difference in the AMPA-NMDA ratio between groups. This suggests that LTP was induced selectively in the CS+ pathways, but not in the CS- pathways, in discriminative fear learning. Our results suggest that the formation of fear memory associated with a specific auditory cue requires selective synaptic strengthening in neural pathways that convey the auditory signals to the amygdala. We next determined whether input-specific LTP is necessary for fear learning by examining how depotentiation, the reversal of LTP, affected conditioned fear to the CS+. After fear conditioning, the mouse showed freezing behavior to the CS+, which was accompanied by synaptic potentiation in the CS+ pathways. We then applied low-frequency photostimulations to the CS+ pathways to induce depotentiation of these pathways in vivo. After low-frequency photostimulations, the mouse did not show freezing behavior to the CS+, indicating the suppression of conditioned fear to the CS+. These results suggest that the formation of discriminative fear memory requires input-specific LTP. Our study demonstrates that input-specific LTP contributes to the specificity of fear memory, enabling adaptive fear responses only to the relevant sensory cue. Our findings also provide important insights for developing a novel approach to attenuate pathological fear in PTSD while preserving adaptive fear memories.